So Carrie and Meredith and everyone else, just so everybody has a heads up, what we're doing right now is we're going to run off the seep for a little bit so we can see how the fauna changes as we approach it from about 140 meters away. We're going to be collecting push cores at intervals on the way there, about 140 meters and 50 meters, and then getting close to where we push cored before. And then we'll go poke around the seep some more to see if there's other good areas to collect some push cores and slurps. We're going to go a little bit past 8 a.m. today, probably till 10 a.m. Pacific time, and then another weather system's coming in, so the ship will probably be shut down from about noon today until noon tomorrow. And then we'll try to get a couple more dives in before the end of the cruise. So that is our current plan and outlook.
The front row
Great.
also thanks you for that update. Good to know. Yes.
So Andrew, I was going to ask, are you taking pairs of cores at each point, or what, a single core, or?
We're taking pairs of cores at each point, and we've actually found that we can get a pretty good mix of kind of all of the different sampling things with just 2 cores, and so we've been taking the microbial samples with the metabolomics / D&B / natural products cores, or subsamples for you, along with rare earth elements. So they're going to be coupled from within a single core, which I think will be even more powerful and interesting.
Perfect, exactly. Yeah, that would be great.
So we'll be doing that, and then the second core will be for probably fauna, so we can see what critters are there. And then when we get closer, we'll do a lot more chemistry.
Yeah. And can you remind me, is there a difference between the bubbling seep, the bacterial community at the actively bubbling seeps versus something like we just saw, the older carbonate areas?
There's a little difference in the microbial community as a function of seepage rate, but most of the seepage rate is really looking at microbial mats that aren't bubbling actively. I think the bubbling active sites tend to act differently, but slower seepage tends to have ANME1s, whereas faster seepage tends to have ANME2s, and then cold seepage has ANME3s. Each one of those is a different microbial group that does the anaerobic oxidation of methane. And then mud volcanoes tend to have, which are much more rapid and vigorous, tend to have more aerobic methanotrophs because there's oxygen mixed into the sediment. So we think with bubbling areas, we get more of the aerobic and different shifts with that. So there are differences across the board.
Great. Hopefully we will see that reflected also in the metabolomics.
Yes. It's also very difficult to get samples from actively bubbling sites because they tend to continue to bubble and launch themselves off the vehicle on the way to the surface. So we try to avoid the really actively bubbling sites.
That makes sense.
Such a contrast, this site compared to the site we dove at yesterday, which was at the same depth but in a canyon, and you couldn't pretty much look at the sediment without seeing a brittle star. And here it's just not that much.
Yeah.
Still lots of pock marks. Lebenspuren, as they call it in the deep sea, tracks of life.
And adjust heading to 050, keep speed and keep tracking. Roger.
one of my favorite deep sea critters that I've never seen because I haven't worked deep enough with a submersible, is a sea urchin that looks just like a nose, like a furry nose, and it buries through the sediment. And the videos of it are just hilarious, and it's time-lapse because they're very slow, but it's just like this random brown nose just digging through the sediment and eating mud.
That's great. I want to see one now. What is it called?
It's something like nostrus, or it has some sort of nostril name in the Latin. It was well named. We'll just put it that way. A friend of mine did his entire dissertation studying them, digging through the mud, and he just wanted to giggle the entire time he was defending his thesis.
Andrew, you're sitting at Psi 1 using that comm panel, right?
Yes, I am. I'm using Psi 1.
Thank you.
Oh, sorry, no, I'm using Psi 2.
Psi 2, okay.
Psi 2.
Thanks.
What's that little thing? Oh, that's another fish. Thorny head. I do like that the fish IDs are so straightforward here, except for that one, which I don't know what it was. Because it's pretty much all thorny heads. I think that was a McTophid. Carrie, were there any other types of samples that you saw that would you'd like us to try to collect on this dive?
No, I think that the mats and the sponges were great, and
Excellent.
yeah.
Got about 120 meters to go to get her kind of over that spot. Roger.
Some more tanner crabs. Or tanner crab, and then there's some nice sea pens coming up.
Nice. Hagfish?
No, that's a coral.
Oh, okay.
That's
I see.
cool. Question
Right.
mark coral.
Question mark. Is that a coral or was that a sea pen? Might have been a sea pen. Portolicids are the elongated bottled shaped urchins. According to Chris, that's not the exact ones I was looking for. Echinocripus rostrata is the name of the nose-shaped urchin.
Man, I could not find anything by searching nose urchin.
I found a YouTube video by my friend who studied it, which was, thankfully, being the person he is, he had the scientific name and the YouTube description. I'm pretty sure that did not help his likes, but
Octopus underneath us there. Bubble.
So the holes, are they from crabs or?
the holes on the seafloor are mostly different worms,
In the sediment. Oh yeah, of course. Right.
and there's also some more of the sort of worm-like phyla that are also pretty common vermiforms, which are not phyla. I mean, it's a diverse group, but lophophorates, things like that. Echurans.
Can you give the name of the nose sea urchin again?
Yes, Echinocripus rostrata. I'll put it in the chat.
Yeah, I was going to say.
It's going to be funny every time you say it.
Wonder how that shape evolved for digging.
Yeah.
I just wonder why it's the same shape as our nose.
Right.
All very interesting questions.
Why nature? Why?
The benefits of a nose morphology.
So a little bit earlier in the chat, Meredith was saying that the curled, what we thought was a coral was actually another sea pen,
Oh, okay.
and it was Anthoptilum grandiflorum. And then the taller, long sea pens are Halipterus species. Also, here's a skate. Another hidden one.
Oh, there's another one there, isn't it? No, maybe that's an optical illusion. Never mind.
Crispin Little said, after looking up this nose-shaped urchin, I presume, if your nose looks like that, then you've got problems.
Seems like for every one of these sea pens, there's an ophiuroid.
Made it look kind of like a telephone pole to me.
All right, I'm gonna stop tracking and do a 20-meter step 020 to kind of bring you over that point.
Sure.
Bridge nav. Can we stop tracking and get a 20-meter step bearing 020? Thank you.
One of those difficult questions to answer. Well, let's see. What day of the week is it?
It's totally Monday because yesterday was Ice Cream Sunday.
Oh, that's right.
Everything's based on that.
An important waypoint.
Like the only day I care that I have to have dinner relief is on Sunday.
Do you only get ice
Deep
cream
sea
once a
salt?
week on the ship?
Yes. Yes, Carrie, we only get ice cream once a week on the ship. It's a hard life.
Plenty of other things to eat.
Yeah. We're surviving. Ooh, big jellyfish.
We have to make do with baked goods the other 6 days of the week.
Oh yeah, cream puffs and cookies and cheesecake.
Oh, it
So-
sounds terrible.
I'm trying to get us to move north a little bit. Your point's kind of gonna be like here-ish.
I'll flip around to it.
We're honestly probably good enough. What do you think, Andrew?
Yeah, I totally think good enough.
All right, I'll hold position and then we could set up for what we need to do.
Sure.
Bridge nav, hold position.
Pretty.
All right, Argus will continue to swing in, but we're good.
Sounds good. I think I'm gonna spin my head around so that I avoid a tether wrap. Like, is that a line there actually?
You saw a line. What's that?
I think it's a C-pen that fell over.
Yeah, looks
Collapsed C-pen.
A sea pen that has had enough.
It's done.
Goodbye, cruel world.
He's just napping. He's fine.
And we're gonna go like a 270. Is that the?
What was that?
Said we're gonna probably go like 270.
When we start going again?
Yeah.
Yeah, ish.
Okay.
Ish. 265, 270, somewhere in there.
Roger that.
Oh, I see daylight out there. Seas look pretty good still.
Yeah.
Yeah, it's supposed to. It should be fine till noon, but a little bit of buffer is not a bad thing.
Yeah.
Sure.
So as soon as we get situated, if you can set down and take a couple push cores,
Sure.
that would be wonderful.
Anywhere around here?
Yep. So these are supposed to be randomly located, so if the pilot will need to close her eyes during the...
My goodness.
Interesting.
Will you will you be able to tell from the analysis later that it wasn't that she had her eyes open or not?
Actually, I'm pretty sure we will be able to.
I'm assuming non-fluted?
Yeah, so let me think for a second. How many push cores do we have, considering we can't use any on the starboard side? So
Yep.
Let me check.
Should we bump the
8.
we have
8 and half of those are the ported cores.
gonna bump out the toolbox a little bit? Awesome. So do you
Okay.
want 1 here or 2?
Let's go ahead and do 2 here.
Okay.
And they can be ported or not.
And Susie, to remind me, only the
Yeah.
top 2, the red and the wooden ball, have been sampled?
Yes, and then also the 2 that are separate outside the crate, but besides that, the back 8 are good.
Roger. So this one here?
Yes.
Alrighty. I need to come in a bit more. Yeah, it's just the shoulders being a bit touchy. All right. Coming up. Some
The eternal question, how long will a rubber band hold? Place your
Yeah.
bets, everyone.
My grip's not very good on this guy.
Or it could be one of those.
Wow.
Oh,
That's
what?
ridiculous.
There we
My
go.
That just went
goodness.
to another planet. Shut
Yep.
up.
Phew.
And really, anywhere you want to put that is exactly the right spot for it.
Copy.
No spot is the wrong spot.
Try to get this as clean as I can for you guys.
Great.
Yes.
Ooh, that's hard.
Is it really?
might be as far in as she wants to go.
Okay.
Oh, maybe she's creeping a bit. Oh, jiggle, jiggle.
That should be plenty right there.
Okay. You want a picture of it?
Yeah, zoom in, please.
I'll go ahead.
All right, cool. Thank you.
Totally. All right, go ahead and come wide. So,
Seems like everybody got really quiet.
We're all focused on coring.
ugh.
These will be presumably samples number 78 and 79.
Won't type 79 yet, it's bad luck.
Yeah, I understand.
And you never hit enter before it's in the box.
Mm-hmm.
What do I not know about 79?
What was it?
What do I not know about 79? The bad luck?
Oh, just putting a sample number down, like presuming it will be okay.
Oh, okay. I thought there.
Yeah, no.
no matter what the sample number.
Oh yeah, I'm 100% with you there. I just thought there was like some reference to the number 79.
no,
I was
no.
like,
No.
no, many numbers that are bad. 2020, for example.
Oh, yes.
That was sticky, huh?
Yeah.
Yeah.
Nice score though. Should have said that out loud.
It's a terrible core.
It's not really.
Oh, this is going to be messy. You ready for your viz to be included?
I thought it was like the 13th floor.
Oh. Yeah, I was going to hold. Yeah, it's not the prettiest right now. One, two.
I thought
Nice.
like the 13th floor, there was something about the 79th core.
No, it's going to be the best one yet.
Best
Yes.
one ever.
Okay, that one's done. One more?
Yep, one more, please.
Sure. All right.
Andrew, can you remind me?
Carrie, I think you cut out there.
Oh, yeah. When would you absolutely use a ported core?
So we use the ported cores if we're going to take pore water out of the side. So we have these little filters that we stick through the side called rhizons, and then we can take the pore water out and analyze the sulfide and sulfate and ions. And out here, the profiles should be really uninteresting. They should be pretty much flat. But when we get close to the seep, there'll be a lot of variation in the concentration of the various ions going vertically, and that can really inform what's going on in the core. So we'll always use those in seep sediments if we, at least in one of the clusters, so we can look at especially sulfide and sulfate, because that transition of where it goes from sulfide to sulfate is very indicative of the seepage rate.
Oh, I see. Thanks. Good.
And the rhizons are really nice because the other option is to bring a giant centrifuge that works at sea and spin out the pore water, or use a pressing system, which is also time consuming and just not fun. So these are really high tech, low tech that work, because it just takes a core with some holes drilled in the side, a syringe.
Push in there, please.
Yeah, can I get a zoom?
Yeah, go ahead, Al.
Thank you.
Sure. Go ahead and come wide.
Nice.
That is a core for ya.
Beautiful core.
Yeah. Something more like that, hey? Use a blind tap right now.
You should put a few more things over the top of that hole, make it even more challenging.
Yeah, I think if you turn it back around, I don't mind doing it backwards. Yeah, do you wanna? Yeah, would you mind rotating the camera 180? Go backwards. Thank you. Good thing all this sediment is consolidated. Yeah. So the only issue is that the shoulder keeps running away a bit. So let's try again. Yeah. I'm gonna reset.
It looks promising, for sure. Nice persistence, Jess.
Woohoo.
Do do do. All right.
Cool.
Sweet.
Good job.
Yeah, it's interesting how it's just doing its own thing. All right. So we're gonna start going towards site J there.
Yep.
Roger that.
And if we can set a point in between there, which is about 50 meters away from point, well, the sampling location.
So 50 meters off of these
Yep.
here? Okay.
Oh, and before we totally go, if we can also fire a Niskin here,
Yeah.
please.
Okay. So we have Niskins number 5 and 6 remaining. So we can do 5
Sorry,
now.
that's me. On.
And then you want it at 1 meter again, or do you want it here?
Here is fine.
Okay. Roger that.
So Andrew, just keep in mind after this, we only have 1 more Niskin.
Yep. Thanks for letting me know. We're going to take that at 10 meters off.
Yeah, okay.
Not a big fan of how it drifts. All right, and we want
Number 5.
number 5. That looks green.
I think so.
Anyone see number 6?
6 is white, I think.
Oh, I see. 6 is flopped up.
Yeah.
Gotcha. Excuse me.
It's in the brow as well, for what that's worth.
Better, thank you. All right.
Not quite.
Swing and a miss.
Yeah.
Better.
Sit.
All right, and
Oh,
fired.
perfect. That's sample number 18, as in 80.
Sorry, 080.
080.
Got it.
Look at that, run away. All right.
Lovely, lovely job.
Sure.
That's fun. Yeah. Thanks, Kim.
All right, we're going to be going 262 for about 110 meters.
262, Roger.
This is pretty decent approximation of where you're heading.
Yeah.
Digging that. Let's go. Like it.
Bridge nav. Can we have a 50-meter step bearing 262? Yep, thank you.
Seeing quite a few hagfish. Unless it's something else. I have legs. Not a hagfish then, I guess. Sea cucumber? Okay, that one I think is a hagfish because it's all curled up.
Which one are you looking at now?
Right above the cores right now.
Oh, the circly spiral shape.
And I think you can see his little creepy eyes.
Creepy face.
Yep.
Are you referring to the hagfish?
Yeah. Creeper.
Yeah.
Oh, cute hagfish.
Bridge nav, another 50-meter step, same bearing.
Like kelp hold fast. Or this one.
Happy little pile there.
Yeah.
A little
It's like
Are those sponges?
it's like a little terrarium or something. It's so
Yeah.
cute.
Can we zoom in?
Can I do a little partial zoom there?
Yeah.
See who's on.
That is adorable.
Aw,
Some anemones. But are the
sponges on it.
white ones sponges?
White ones I think are nudibranchs, right? Okay, go ahead and come mine there, Al.
I mean like the creamy white circle ones.
They certainly look like the buttony sponges.
Yeah.
No, thank you.
little skate there.
Just a slight adjustment to 270.
Roger.
Ah,
So Andrew, are you going to pull the full 2.5 watches? You going to make it?
I'm hanging in there.
remains to be seen.
Seeps have been amazing. Just spectacular.
Oh, it's
Really.
a tiny little
Best
skate.
dive ever, Nicole.
No, it's been the exact opposite. We've had a variety of, so big carbonate outcrops on the surface. First thing we hit was a nice microbial mat, which we sampled initially. We've got 2 worms. For the OCNMS folks, there were a couple of corals that they were really hoping to get that they hadn't seen yet, so we got them for them. Our Niskins are almost full. Now we're just doing a transect on, and then we'll probably come up over the rise.
We got a few sponges too. We're heading towards the seep now.
Yeah, so the seeps are all up here. It's a big hump. Yeah, big carbonate hump. Big blocks with valleys down beneath, crabs all over it. We're doing a distance transect. So we took some sediment cores off here, then we'll take some cores as we get closer. Yeah, we pretty much stumbled straight across the seep after we got in the water. And there's just little pockets where it's clearly very, very, very active. It's just not bubbling right now. Don't see any. I think probably 9 or maybe 10. 9 or 10. Looks like we're just starting to get back into it.
Yep.
Great.
Bridge nav, hold position.
Good, thanks.
All right, I think you should be able to creep into that core site. That's what we're doing, right, a core?
Yes, please.
Maybe another 10 meters.
looks like we're just about.
Yeah, I think that's
Mark.
pretty solid. We're pretty close.
Yeah.
So do you want it anywhere around here then, Andrew?
Sorry, was that a question for me?
Yeah, do you want the core anywhere around here? It looks like we're pretty much on target.
Yeah, this looks